going to produce a sperm cell that carries a brown allele. It's what?
25.
It's close to 25%.
50.
It's 50%, right? He can produce two types. He can either produce brown or he can produce green. And the probability of producing brown is equal to that of green, right? Because it's just random chance events. So 0.5% chance or 50% chance that dad's going to produce a brown-eyed sperm cell, which is crazy to think about, right? And there's a 50% chance that that's going to carry the green allele. It's the same thing for mom. Mom, there's a 50% chance that she's going to produce an egg cell that carries the brown allele and green allele. So then what's the probability that they will have an offspring that is brown-eyed? What's that?
25%.
25%. It's 25% because we know that there's only one of those, right? 4 squares, only one of them is brown-eyed. But mathematically, how do we calculate that probability? The way that we do that, does anybody know? We find the product of the probabilities, right? If the probability of a brown-eyed sperm and a brown-eyed egg cell are each 0.5, we just multiply those two probabilities. So 0.5 times 0.5 is 0.25. So this has a 0.25 probability. Does that make sense?
Well, it's kind of like in math when you're first learning multiplication, they make you essentially a Punnett square that you do your multiplication with. So you just use that same technique.
I wish that I would have used a Punnett square in math, but I never did. But if that works for you, great. It's the same thing for a green-eyed individual, right? The probability that dad's going to have a green-eyed sperm and mom's going to have a green-eyed egg are 0.5 apiece. And so we take 0.5 times 0.5, and the probability is 0.25. What's the probability of a heterozygous or a hazellite offspring? What's that? It's 0.5, but why? Because the probability of each one of these is what? 0.25. And there are 2 of them, so we just add those probabilities together. 0.25 plus 0.25 is 0.5. Okay. We okay with that? What's the probability of producing an offspring that carries at least one brown allele? 0.75, because this one does, this one does, and that one does, right? So it's 0.25 plus 0.25 plus 0.25, which is our 3-to-1 ratio, right? That's 0.75 to 0.25, which can be converted into 3-to-1. Okay. So the question is, what if there is an individual, and let's go back to flower color really quickly. What if there's an individual that is purple? What are the possible genotypes for a purple individual if that purple character is dominant? Yep, we could be homozygous dominant, or, yeah, exactly right. How do you know what genotype that individual actually has? Doesn't that seem like a problem? So let's think about this, you guys. In humans, there is a genetic disorder called cystic fibrosis. And cystic fibrosis causes mucus to build up inside the lungs, and often children die before the age of like 8 years old. It has a very high rate of mortality. It's a recessive disorder. So your potential mate could carry that recessive disorder. They wouldn't necessarily demonstrate it, right? Because they have a dominant allele, but they could carry it. So wouldn't you want to know if there was a chance that your potential mate is with you going to produce a child with cystic fibrosis? Doesn't that seem like a good thing to know? So how do you figure that out?
Family history.
Family history, that's a really good way to do it. Look back through your pedigree, see if there's any case of cystic fibrosis. But you know, as a product of probability, that family lineage could carry it and never express it because it's recessive. How else could you do it? Could you think of a potential cross between, let's say, plants, where you could tell whether or not an individual is homozygous dominant or heterozygous? What's a cross that we could do?
Mendel just made another generation to see what it was, but that's probably not a good idea.
Yeah, that's exactly what he did. But what would he have crossed that with? What genotype would he have crossed those individuals?
Lowercase p and a lowercase.
What's that?
Lowercase p and a lowercase p.
Exactly right, homozygous recessive. So really there are two options, right? We could either cross that with a true reading purple individual or a true reading white individual. But the only one that's going to work is a true reading white individual. I'll show you why. So if we take this and we cross it with that, that's going to come up with a very different phenotypic ratio than that. What would the phenotypic ratio and the genotypic ratio be for that cross? They would all be heterozygous, right? So that would be a ratio of 1. And we know that's true because this is what Mendel did when he crossed his first cross, that parental generation. True reading purple, true reading white, all of the offspring were what? Heterozygous. Right? Doesn't that seem to make sense? So if this individual is purple, which we know it is, and it's homozygous dominant, then what we expect is from a test cross, that's what this is called, all of the offspring to be purple. Alternatively, if this individual is heterozygous, expressing that purple allele but carrying a white allele, what's the phenotypic ratio?
It's 1 to 1.
It's 1 to 1, right? It's this one again. Right? Test crosses allow us to figure out what the genotype is for an individual that expresses the dominant phenotype. This is the test cross.
So it's just like plugging that in, kind of. Just like, like let's say, like for the PP, you're just plugging that lung pheno. Just saying. Oh.
Yeah, I mean, you really wouldn't want to do this with humans, right? I mean, you wouldn't want to take your potential husband or father of your children and say, I know this person has cystic fibrosis, so go ahead and have an offspring with that individual to see whether or not you actually have cystic fibrosis. You wouldn't necessarily want to do that. But for other organisms, you could, right? Plants, that works well without violating any sort of ethical standards. Okay, does this idea of a test cross make sense? Okay. So on the exam, you guys, I'm going to present you with genotypes and ask you what the phenotypic ratio is going to be, or what the genotypic ratio is going to be. Or I could say, there is an individual that expresses the dominant phenotype. And you cross that individual with another that expresses the recessive phenotype. 50% of their offspring have the dominant phenotype, and 50% have the recessive phenotype. What was the genotype of the parent with the dominant phenotype? It's heterozygous, right? That's this scenario that we've just described right here. Okay. So this is, again, a test cross, and this shows you what Mendel did with his first and second experiments, right? I guess this isn't a test cross just yet. This is true reading purple with true reading white in a Punnett square. All of the individuals in the first filial generation ended up being purple, and all of them were heterozygous. So there was a phenotypic ratio of 1, and a genotypic ratio of 1. They're all the same. If we take those two individuals, or two of those individuals, from that F1 generation and we cross them, we're going to get our 3-to-1 ratio, at least phenotypic ratio, right? And 1-to-2-to-1 genotypic ratio, and then this is the test cross. This individual is purple, but we don't know if it's homozygous dominant or if it's heterozygous. If it's homozygous dominant, we expect all of the offspring to have the dominant phenotype. And if it's heterozygous, we expect 50% of the offspring to have the dominant phenotype. What if you did that though? What if you crossed 2 plants, you did a test cross, and all 4 of the offspring, and you only produced 4 of the offspring were purple? What would you assume?
They were homozygous.
You'd assume that it was homozygous dominant, right?
Yeah.
But what if I told you it's actually heterozygous? Would that surprise you very much? And I would hope not, because it's a problem of sample size, right? If we crossed them 1,000 times and all of them ended up purple, we could with some degree of certainty say, yeah, that's got to be a homozygous dominant individual. I guess the point I'm trying to make is there's an issue of sample size that we need to be concerned with as well. Okay, so from that experiment, Mendel came up with an incredibly important law. Now, laws in biology are few and far between, but this is a law, a scientific law. It's called the law of segregation. And what the law of segregation states is, at the point of gamete formation, those 2 pairs of genes, those homologous genes, segregate into different gametes, so that each gamete ends up with just a single copy of each gene. That's the law of segregation, i.e. meiosis. Isn't that rad? I mean, Mendel, without looking at cells, without identifying DNA, was able to describe meiosis just by taking a bunch of peas, seeing what happens with them. There was a second law that he described as well, and that's called the law of independent assortment. And he described the law of independent assortment by looking at 2 characters simultaneously. And we're going to need some board space for this Punnett square because it's rather large. I'm going to erase this side over here if that's okay. Okay. So remember, Mendel did this experiment not just with flower color, but he did it with 5 other characters as well, right? Pod size, or excuse me, pod shape and pod color, seed shape, seed color, and then also plant stature, how tall plants actually grew. Because there was a dwarf variety and there was a variety that had normal stature. So he wanted to know if genes truly were independent of one another. That is, if there is no cross effect and if genes could be inherited independently. So the way that he did that was he looked at 2 characters simultaneously. And the first character that we're going to say he looked at was, again, flower color. So we know that the gene for flower color has 2 alleles. It has a capital P allele for purple, and it has a lowercase allele, or a recessive allele for white, right? Now, there's also variation in seed color. Seeds are either yellow or white, or excuse me, yellow or green. And our assumption is that the dominant is green, because that's the color of the peas that we eat, right? But actually the dominant color is yellow. And so the gene is seed color, and the dominant allele is yellow, and the recessive allele is green. Okay. So what Mendel did was he took individuals that were heterozygous for both genes and he crossed them. That is, he took an individual that was heterozygous for flower color, capital P, lowercase p, and that same individual that was heterozygous for seed color, capital Y, lowercase y, and he crossed that with another individual that had the exact same genotype. That. Okay? So this is called a dihybrid cross. This is a monohybrid cross, mono meaning one, and this is a dihybrid cross, di meaning two.
It's called monohybrid cross.
Okay, so how do we do a Punnett square for a dihybrid cross? Well, instead of a 2x2 square, we're going to have a 4x4 square. Okay, this is where things get a little bit tricky. And what you need to remember is that the head of each column and the head of each row represent a gamete, and a gamete has to have a copy of each gene. So let me start you off. What we can say, right, is that Dad can give up a capital P. He can give up a purple flower allele. What are we going to pair with that? Lowercase what? What if we did that? What if we put another P here, and we did the same thing for Mom? What's that offspring going to have? How many genes does it have for flower color? 4. How many should it have?
2.
How many genes does it have for seed color?
None.
And how many should it have?
15.
2. So this can't be right.